Let's understand the very basic details of sensory physiology in this lecture. What are sensory receptors? These are specialized cells. They could be neurons, they could be epithelial cells. They have the ability to detect a change, whether it be within the body or changes that are occurring around us. They're sensitive to one specific change, and when there is change, such as in this illustration, this is chili pepper, the chemical that's in here, capsaicin, is the stimulus. If this is a neuron that's responding to the chemical in the chili pepper, how it responds is by going through changes in its membrane properties. Ion channels are open, and specific ions make their way into the cell most of the time, and it's typically sodium ion entering the cell, causing depolarization. Now, the depolarization that you see in a sensory receptor is what you call as receptor potential. It's a graded potential. So the strength of the stimulus would determine the shape of the curve, the amount of ion that enters the cell and causes a depolarization. And if the depolarization is sufficient enough to generate an action potential, you refer to that as generator potential. And ultimately, it becomes an action potential. So a sensory receptor has the ability to detect a change and then convert this signal to the language of action potential. And this process is what we call as transduction. Transduction is the characteristic of a sensory receptor that detects a change and then convert it into an action potential, whether it be a chemical change or the photoenergy or a mechanical stress, temperature change, or a pain, touch, pressure, olfaction, taste sensation. All these changes are simply converted into one common form, the action potential. That's the unique nature of sensory receptors. What is the difference between sensation and perception is what we want to understand here. Sensation, as I mentioned earlier, is the ability to detect a change. This typically happens at the level of sensory receptor. Okay, sensory receptor is designed to respond to a specific change, so it goes through a change such as we saw before, and channels open, depolarization happens, and it becomes an action potential. And it's transmitted to the brain areas that are specialized to sense this change, detect this signal as meaningful. And perception is where it's the understanding of the action potential. So it's not only the action potential, but maybe things associated with it. If you were to taste a particular food, it may be combined with your previous experience of how tasty it was or who you ate with. All those things combined basically to be of a meaningful message. Perception is basically realizing the change. Understanding the change is perception. Sensation is basically the change is detected. Okay? Now, we will talk about the different types of sensory receptors. Classification of sensory receptor is really complex. There are so many different ways by which you can organize sensory receptors. If you see sensory receptors that detect just the changes outside the body, then you can call them as exteroceptors. If there are sensory receptors dealing with changes within the body, such as oxygen levels or hydrogen ion concentration, etc., they could be called as interoceptors. And sometimes the classification really overlaps. And one other form of classification is based on the structure. That's what we have here. So let's go over it. The first one that you see here is a very simple type of sensory receptor. You can recognize this as a unipolar neuron. That's one cell body, one extension that divides to become two extensions ultimately, one serving as dendrite and then the other one serving as axon. This is a simple free nerve ending serving as a receptor. Okay, pain receptors, thermoreceptors are good examples of free nerve endings. Now, what we see here is a unipolar neuron. Okay, and what happened here is the dendrite part of the receptor is modified, is simply encapsulated, meaning a connective tissue coat around the nerve ending is what we see. So it's a little specialized, and that's again a general sensory receptor, or both of these are general sensory receptors. This could be a tactile receptor detecting touch or pressure. Okay. Now, let's talk about a specialized sensory receptor. Note here that it is a bipolar neuron. Here is a cell body, an extension on this side, another extension here. Okay, so this entire structure is one cell body with one extension here, one extension here. This is actually a photoreceptor called rod that you find in large numbers in the retina of the eye. Okay, so please note that here it's not a nerve ending, but it's a very specialized cell, bipolar neuron, and then it's connected to another bipolar neuron and another, and so on, ultimately becoming a nerve that you call as optic nerve. In any case, what we want to know here is there are simple structures, there are intermediate structures, and there are highly specialized structures. You may have learned that unipolar neurons typically serve as general sensory receptors, and bipolar neurons typically serve as specialized sensory receptors. So that's one difference between general sensory receptors and special sensory receptors. What other details can we find here? Well, how do we define one receptor as general sensory receptor? Typically, these sensory receptors are found in more than one location. Okay, general sensory receptors are scattered often in the skin and maybe the lining of the bone, maybe the lining of the blood vessel, etc. You see, whereas specialized sensory receptors are limited to one special area, which is typically well protected compared to general sensory receptors, which are scattered, and for example in the skin that's exposed to all kinds of hazards, chemical hazard, mechanical hazard, etc. It's not well protected, you see. So general sensory receptors less well protected compared to special sensory receptors. General sensory receptors are found in more than one location. Special sensory receptors are limited to one main location, and then unipolar neuron in the general sensation versus bipolar neuron in a special sense. Yet another key difference is that general sensory receptors carry their signals to the general sensory processing area within the parietal lobe that you refer to as somatosensory cortex. Whereas spatial senses are typically dealt with by specific lobes, auditory cortex for processing of sound and understand all about hearing, or a visual cortex for processing the photo energy. So those are some differences between general and spatial sensory receptors. All right, now let's talk about yet another way to classify sensory receptors. This is based on the nature of the stimulus that they respond to. Mechanoreceptors detect mechanical change. It includes stretch receptors, osmoreceptors, and so on, okay? And typically they are encapsulated nerve endings. Thermoreceptors, they are free nerve endings. These detect change in temperature. In fact, you have two subtypes of thermoreceptors: warm receptors and cold receptors. So temperature of a certain range by cold receptors, and another higher range for warm receptors. And nociceptors are also free nerve endings. They detect damage. Okay, they're also called as pain receptors. Nociceptors actually detect more than one type of stimulus: mechanical stress, chemical stress, thermal stress. Because they can detect more than one type of stimulus, they're referred to as polymodal. Okay, now the electromagnetic type of receptors, they are the photoreceptors, rods and cones. They detect photo energy or light. Chemoreceptors detect chemical changes. Olfactory cells or olfactory receptors detect molecules that are present in the air, which ultimately dissolve in the mucus. Our chemicals that are in the food are detected by the taste receptors, taste buds. Similarly, levels of CO2, levels of hydrogen ion, levels of oxygen ion are detected by chemoreceptors in the lining of the blood vessel or in the brain regions, etc. Okay. With this, let's move on to look at the sensory receptors in a very classic example, a sensory structure, the skin. It houses a number of receptors. It houses free nerve endings, which serve as pain receptors, there are thermoreceptors. It also houses a number of tactile receptors, the corpuscle, the root hair plexus. Then you're talking about Pacinian corpuscle here, and then the Merkel disc or Merkel cell, and then the tactile corpuscle. So there are a variety of these, and not all of them are present in all parts of the skin. Fingertips may have these fine touch receptors or these deep touch or pressure sensitive receptors, and so on. And even in the skin, there are certain parts that are very, very sensitive: lips, fingertips, the nipple area, etc. So those are the ones that are concentrated in certain types of tactile receptors compared to some other parts of the skin. All right, now let's talk about a few more characteristic features. A modality, what is that? A sensory receptor is optimized to detect typically one specific type of stimulus. And how does it know to detect just one type? It would have a low threshold for that particular stimulus. Low threshold for that particular stimulus. That's how it can respond to that change more readily than for other stimuli. Sensory adaptation, which we will discuss in the next slide, is the way some sensory receptors respond to stimulus. Some sensory receptors continue to respond to a stimulus as long as the stimulus is maintained. Some other receptors, on the other hand, would respond to a stimulus initially, but if the same stimulus stays longer, these receptors stop responding to the stimulus. Only when you change the strength of the stimulus, they respond again. So they are ready to ignore the stimulus as long as the same stimulus, same strength of the stimulus is maintained. Receptive field is the area a sensory receptor deals with. For example, in the skin, say the fingertip, you have touch receptors that are given a specific area of the skin to deal with. So you would expect that the skin area, a touch receptor in the fingertip handles is going to be very, very small compared to the receptive field a sensory receptor, a touch receptor would have in the locations such as the neck or the arm or the trunk. Okay, so larger the receptive field, less sensitive the sensory receptor would be, or less detailed the information would be, not so precise, in other words. Whereas in the fingertip, because the receptive field is very small, you will detect fine touch sensation. Okay. The other characteristic feature is labeled line principle. What this deals with is that the sensory receptor is not just one sensory receptor taking the stimulus signal in the form of action potential directly to the brain area. It cannot. So a sensory receptor could be a nerve ending, but it typically synapses with another neuron and maybe yet another neuron. Okay, in that situation, you would have one originating at the sensory receptor referred to as a first order neuron. Then the one that it synapses with next is called the second order neuron. The one it synapses with later is called third order neuron. So first order, second order, third order neurons is how the pathway is constructed. Okay, so this is what you call as labeled line. So this is for the transmission of the sensory signal. Okay, so the stimulation of this particular label line would be sensed as bringing a specific signal. So it's automatically assumed that a particular label line is dealing with this particular sensation based on previous experience and so on. And another interesting thing is you can activate the label line in any part of the pathway. Okay, whether it's second order neuron or the third order neuron or the first order neuron that you stimulate, it would still be perceived as the stimulus coming from that region. So that is an interesting characteristic feature of sensory receptors. Adaptation is shown here in detail. Note here, this is rapidly adapting or phasic receptors. So you're looking at the stimulus being maintained continuously, and what you see is a receptor potential which becomes an action potential. You have about 3 action potentials generated in response to the initial stimulus, but when the stimulus is maintained, they stop responding to this stimulus. Pacinian corpuscle is an excellent example of that. There are some touch receptors that would do the same. Now, slowly adapting sensory receptors or tonic receptors, as you can tell, if the stimulus is maintained, there is an initial burst of signal here, more or less like that of rapidly adapting. That's only in the initial period. There are more signals generated, more action potentials generated, but as long as the stimulus is maintained, it continues to respond, but they're far apart, and you notice that. But it can be a long-term response. So slowly adapting or tonic receptors keep brain appraised of the status of the body with respect to its surroundings. It may take hours or days for it to give up responding to that stimulus. Muscle spindle is a good example. Golgi tendon apparatus and Ruffini's endings, Merkel's discs, macula, chemo, and baroreceptors. They're all tonic receptors. Most of these are protective receptors. So in order to protect the body, they are designed to continue to respond to a stimulus so that a specific action can be taken eventually. Well, we'll stop at this, and in our next lecture, we will specifically talk about one type of sensation, that is pain. Thank you.